Happy Thursday morning. You know what? We made it to class number 3. That's a great achievement for us. Let's take a nice deep sigh of relief to celebrate. Class number 3. Today's subject will be, we're going to review basically all the network motifs in E. coli transcription network. We'll cover everything you need to know by the end of this class. Everything you need to know in order to calculate the dynamics of the entire E. coli transcription network. Do you believe it? Can we do it? Can we take another deep sigh of relief? Will you go on this journey with me? Okay. So first things first. Let's review what we did last time. Let's take another nice sigh. Thanks for giving us a chance to take a sigh of relief in your honor. While I'm erasing, try to think what we did last time. Yeah. Okay. So small review. Last time we looked at our first network motif, a pattern that's overrepresented in the E. coli network happens again and again and again and again. The pattern we looked at was negative autoregulation and positive autoregulation. And we saw that these little circuits can have useful functions. Let's take a nice deep sigh of relief. If this is time and this is X concentration, then just simple regulation without this positive or negative feedback loop has the dynamics. It looks like this. It goes to beta over alpha. It has its famous exponential approach to the steady state. Now, if we make a circuit with the same steady state and the same degradation rate, but we give it a negative autoregulation, it speeds up. Negative autoregulation. But if we give it positive autoregulation, it slows down like that. We also saw that negative autoregulation can stabilize the steady state against the steady state level against variations, noise, changes in beta, changes in alpha. And that positive autoregulation actually has the opposite effect. It increases noise even to the extent of having bistability, which is an all or none gene expression circuit. By the way, who's here for the first time? Welcome. Welcome, welcome. Great. I guarantee you'll be able to understand this. It's a modular design, this class. All right. We're going to go now to look at larger circuits. Before I do that, I want to ask if there's any questions. Yeah?
You said that the negative autoregulation, the steady state is K, right? The affinity of.
So the comment is this: for negative autoregulation with step-like regulation, the steady state is K, not always.
What is the limit of K?
How large can K be, this halfway repression point? So it's in units of concentration. It's a concentration of the transcription factors needed to 50% repress its own promoter. And this could be vary across a range of, let's say, at least 3 orders of magnitude from a few molecules per cell to a few thousand molecules per cell, I would say typically. Cell, I mean E. coli volume. When you think about human cells, you need to multiply things up by a scale of about 1,000. But things do scale in many ways. Time scales also grow by a factor of 10, approximately. So if E. coli has a generation time of an hour, a human cell has a generation time of a day, typically. By the way, you might ask yourself, what's the use of a delay? And today we'll see an example where a delay is useful. So keep it in mind. So I'd like to do now the project of looking at all the possible network motifs in the E. coli network. E. coli network, as you remember, is a directed graph where the nodes are genes and arrows are transcription factor X changes the production rate of transcription factor Y. We look at this graph and we can count all the small patterns, the subgraphs in it, all the self edges like we saw, all the two-node patterns. In two-node patterns, nothing much interesting happens. So three-node patterns, we can count all the possible ways to connect three genes. And I'm going to draw them all. And we're going to look for network motifs, ones that occur a lot in the network. So I'll try to draw all the 13 ways to connect three genes. So here are the three genes, X, Y, and Z. And this is one way. X regulates Y and Z. Now, I'm not going to write X, Y, Z anymore, just the arrows. You can have a gene regulated by two transcription factors. You can have a cascade. Then you can, of course, have mutual situations where X regulates Y and Y regulates X. Why not? And they regulate a third gene. This mutual situation where this gene is regulated, these are different because this node has one incoming arrow, and here this node has two incoming arrows. So that's topologically different. You can have this situation. And now triangles. This triangle and this triangle. They're different because this is a cycle. And here it's two paths converging. It's not a cycle. Let's take a nice deep sigh of relief. Welcome. And yeah, and then we have triangles with a mutual regulation. These three possibilities. Then we can have two mutual regulations in a triangle and the fully connected clique. Okay? Let's count if it's 13. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Whew. Why am I drawing all of these? To make the point that there's lots of subgraphs and possibilities in a graph. If we go to 4 nodes, there's 199. If we go to 5 nodes, it's 7,000. It grows exponentially. So there's a huge amount of possibilities. Yeah, and here I drew arrow. It could be either activation or repression. So if you had the sign, it's more. If you had autoregulation, it's more. So you have many, many possibilities. And here I drew the things without autoregulation, without the signs. Thank you. But when you look actually in E. coli, which ones appear at all, you find in E. coli, find this one, this one, this one, and this one. So only 4 of them appear. Cycle, for example, doesn't. And then if you compare it to random networks, the only network motif that's more than expected by chance is the feed-forward loop. Cycle is an anti-motif. It actually occurs less often than you expect in random networks of the same size. The other ones don't appear at all, and they don't appear in the random networks either because by chance it's hard to get these mutual regulations. So here's a big simplicity. Out of the 13 possibilities, only very few appear, and there's one, you can say, really interesting one, the feed-forward loop, which occurs, let's say, at least 300 times in E. coli, with each time with different X, Y's, and Z's. So we're going to focus on the feed-forward loop. Benjamin, I see you puzzling. High number.
I was calculating how many.
Okay.
Yeah. I mean, there are not so many transition factors, so a lot.
Right. But they have a lot of output genes. That's correct.
Yeah.
That's the.
Okay.
And it occurs more than 10 standard deviations more than 10 than in randomized networks, which are very strict randomized networks where you keep for each node exactly the same number of outgoing and incoming edges and you shuffle them. So we're going to look at the feed-forward loop for the first hour here.
What exactly do you mean when you say network motif and anti-motif?
Network motif is a pattern that occurs many times in the network and more often than expected by comparing to some null hypothesis network, like a randomized network. This p-value in a random network is vanishingly small. An anti-motif means that this occurs more often than in random networks than in the real network. Did I explain myself? Yeah.
Is it that in the random network, the possibilities are distributed amongst all these possible space, and within a logical network, it's restricted because of some, I don't know, some
Correct.
limitations?
In biological networks, it's restricted to very few subgraphs because we argue of evolution favoring some subgraphs because they have useful functions and disfavoring other subgraphs because patterns. Because, for example, this one, I can show you later, leads to these noisy oscillations. It's unstable. And if you want an oscillator, there's a better, more accurate circuit to use, for example.
So in a randomized system, if you place the same limitations, it should behave like the biological network?
So the question is, if you take a random network and you start evolving it on the computer with some goal, should it evolve to have these network motifs, for example? So the trick is to find the right goals. In principle, yeah, there's quite a lot of work on that in the last decade. Let's take a nice deep sigh of relief. So we discovered this idea of network motifs in around 2002. And there's a few thousand papers about it. So of course, I haven't read all of them, and I don't know. People have taken this into other networks, etc. But works like this are definitely interesting. Okay, so we're going to focus on the feed-forward loop. I'm going to erase now. Any objections? Objection certain. We're going to focus on the feed-forward loop. It has two ways to get from X to Z, a direct path and an indirect path through Y. Other way to say it, there's a direct path and a delayed path that converge. In order to understand what it does, we need some more information. Each of these arrows could be plus or minus, activator or repressor. Right? So each one can be plus or minus. That's 2 times 2 times 2, 8 possibilities. There are eight types of feed-forward loops. This one, where all activators, we call coherent type 1, C1, feed-forward loop. FFL from now on, feed-forward loop. Okay? This is the C1 feed-forward loop. I say coherent because both arms are doing the same thing. You activate, and you activate an activator, which is to activate at a delay. And there's 3 other ways to have coherent feed-forward loops. You can also repress an activator. You can also activate and repress a repressor, because minus minus is a plus, right? Repressor, repressor, like activating, so it's coherent. You can also repress and activate a repressor. Okay, these are 4 ways, and we call them coherent type 2, coherent type 3, and coherent type 4. These are the 4 coherent feed-forward loops. And then there's 4 others. For example, X can activate Z, but also activate a repressor of Z. That's weird. It's saying two opposite things. It activates, and it activates a repressor. We call this, therefore, Shmulik Mangan, when he was looking at this, the incoherent feed-forward loop, or incoherent type 1 with this configuration. Incoherent. And there's and other ways to have incoherent feed-forward loop is you can repress and repress a repressor, because minus minus is a plus. This is plus and minus. You can activate and repress a activator, and you can repress and activate an activator. Okay, so this is I2, I3, I4. If you can notice, the coherent feed-forward loops always have an even number of minuses, and the incoherent feed-forward loops have an odd number of minuses, 1 or 3. So we're going to actually discuss both types. Now, I told you that in E. coli, there's about 50-50 plus signs and minus signs. So you could expect, as a first approximation, that all these 8 feed-forward loop types would be present at about the same amounts. But that's not the case. When you look at the types of feed-forward loops in E. coli, you find that 90% of E. coli FFLs are coherent type 1 or incoherent type 1. So again, a big simplicity. Of the 13 possible circuits, only one is a network motif, and of the 8 types, only 2 are found. And the 10% is, the other 10% is distributed between the other feed-forward loops. In human cells, you find more of these other types, alternative types. Sometimes the Y, when it's a repressor, can be a microRNA, for example. That's a big network motif in mammalian cells. So we're going to understand the coherent type 1 and the incoherent type 1 feed-forward loops. We're going to start with the coherent type 1 feed-forward loop. Another thing we need to know, there's 2 transcription factors. So look at this. These are all activators. X activates Y. 2 activators together activate Z. Just 2 activators. How do they work together? Okay. So perhaps I'll draw the situation here. X, the activator, becomes active-inactive on a rapid millisecond timescale. It has a signal that pushes it to be active. When it's active, it binds to the DNA of gene Y to make protein Y. So it has its binding site here, X binding site. And Y itself is a transcription factor. It can be active-inactive. It has its own signal. So there's 2 signals to the feed-forward loop. And they combine X. Of course, in the cell, there could be 1,000 molecules of X. So some of them bind here, one of them bind there. And Y bind to their sites. This is the Y site. This is the X site. So a series of letters in the DNA that they recognize. So the question is, so we recognize the feed-forward loop has 2 input signals, SX and SY. It's a circuit with 2 inputs. That's probably its major function. It integrates 2 inputs. How does it integrate them? What logic does it do? So it turns out that a good approximation for a lot of bacterial promoters and non-bacterial promoters is the 2 regulators are combined with what we call an input function. And we're going to analyze 2 flavors, X star and Y star. You need both of them. Both are needed. Both need to bind the Z promoter to activate transcription. And we're going to consider the OR gate. Either is enough, right? So these 2 possibilities. And mechanistically here, biology shows its full complexity. There's many, many ways to get AND gates by having molecular interactions and touching the RNA polymerase in different ways, or OR gates. And there's so many ways. Each gene finds its own way. But that's all on the fast time scale, so we don't need to worry about that for our discussion. We're going to worry everything is fast. The slow reactions, as you remember, are the accumulation, basically, of these proteins. Yeah. So we need to worry about the coherent type 1 feed-forward loop with an AND gate and with an OR gate. Okay? So there's lots of possibilities here. But we're going to do them all. I have great confidence. Yeah. So we talked about coherent, incoherent, and OR. And before I take your question, just want to write down a little biological example here. E. coli can eat a lot of different sugars. Its favorite sugar is glucose. It likes to grow on it even with the other sugars. But when there's no glucose, it's starving, and it opens up the possibility to eat other sugars like arabinose. So this calculation it needs to do is make the arabinose genes if there's arabinose and you're starving for glucose. Make the arabinose utilization proteins only if you're starving and there's arabinose around. The circuit that does this is a coherent type 1 feed-forward loop with an AND gate. The input signals are cyclic AMP, which in E. coli means I'm starving. And RC, which binds to the sugar arabinose. That's an example. One of the hundreds in E. coli. That's why the feed-forward loop, first of all, it does an AND gate computation in this case. But what else does it do? You see, for an AND gate, you don't really need this arrow. You can have two transcription factors regulating a gene with an AND gate. So why do you need this extra regulation? That's what we're obsessing about. All right, so now that we've set up the molecular situation of the feed-forward loop, we deserve a nice deep sigh of relief. And by the way, you can do this when you're working on your computer. You notice you're not breathing deeply. Stop, take a nice deep sigh of relief. You were nowhere, and now you're now here, as Mike Myers says. All right. Yes?
You said that C1 and I1 are the most prevalent.
Yeah.
Could it be because in both of them X is always an activator, it doesn't have to switch function?
Yeah, the comment is in both the common feed-forward loops, X is an activator for both of its genes. And that happens more often than situations where X is an activator for one gene and a repressor for another gene. That's right. That's a factor.
Happen more often because, well,
I don't know why, but certainly a lot of activators are repressors for some gene. But in this case, for example, this is a repressor for both genes. That's normal, but it doesn't happen so much. And actually, you can think about why is it? What's wrong with the other feed-forward loops types? And yeah, it's kind of an open question, actually. But you notice that if I just, for example, move these two, this plus and minus, like this, I get this. And that's very common. So it's kind of sensitive to the signs and the arrows, right? The topology matters. Okay, we're going to focus on the coherent type 1 feed-forward loop with an AND gate and look at its dynamics. So I'm going to focus on the coherent type 1 feed-forward loop with an AND gate and focus on that. So I'd like to do something fun with you to help you get the intuition. So for that, I need 3 volunteers to come up here and do a nice, safe demonstration of feed-forward loop dynamics. So who's willing to come up here? Daphna, Asi, and Yel. Let's give them a round of applause. Don't delay. All right. So just stand here in a row like this. Okay, great. Asi, you'll be X. Okay. Your job is to sense the input signal, SX. And so when I say signal is there, you raise your hand right away. So can you do that? Signal is on. Signal is off. This means that X is active. That happens quickly. Okay. Daphna, you'll be Y. You look at Asi, at X. And when X is on, you start being made, because X binds the promoter. But you don't get turned on right away. You have to accumulate and cross your activation threshold for Z. So you count 1, 2, 3, and then you raise your hand. Let's try it. Signal is on. Count out loud. Out loud.
1, 2, 3.
Great. And when signal goes away, you also decay slowly. Signal is off.
1, 2, 3.
Great. Now you have the hardest job. Gil?
Li.
Gili. You're the AND gate in the Z promoter. So you need both of these to bind. So only when both of their hands are up, you raise your hand right away because your RNA transcription rate. Right away you raise it. All right? Now your job, you also have a job. Observe what happens when there's an on step of signal and an off step of signal. Okay? And then you tell me what happens. All right, ready? Take a nice deep sigh of relief. All right. Signal is on.
1, 2, 3.
Signal is off.
1, 2, 3.
Let's do it again. Okay? Signal is on.
1, 2, 3.
Signal is off.
1, 2, 3.
Okay. Let's give him a big round of applause. Thank you very much. You did very well. So what did you observe? What's the difference between adding a signal and taking away a signal as a function of Z? For Z.
Delay, turn on, and turns off right away.
There was delay when you turn on, and there was no delay when you turn off. Did you see that? So for an on step of signal, there's delay in Z production. I'm going to draw 3 graphs here. One moment. Time. This is the signal. So on step means this. At time t equals 0, I add a lot of signal. That's called an on step. And of course, this you can also say that this is how much active x there is, because that happens quickly in our approximations. What about y? 1, 2, 3. Y starts being made and accumulates. What about z? Because of the AND gate, one arm isn't enough. You need both to bind. That means y needs to accumulate and cross across its activation threshold, kyz. I'd like to put in this number here. This is the concentration of y star needed to bind the y promoter. So this number is important here. You have to cross this number. In the step approximation of step input function, it's all or none. And if it's a Michaelis-Menten approximation, it's gradual. But we're going to make the step approximation. So only in this point, Z starts being made. This means there is a delay. Delay after on step. No delay after an off step.
But the Z production after the delay was immediate.
Well, the production of RNA, Gili was the production of RNA. You were the production of RNA. It was immediate, and protein starts accumulating.
Oh, that's
Thank you. So RNA gets turned on within minutes. Protein gets accumulated on the time scale of a cell generation, as you remember. Did I explain myself in this demonstration? Yeah, I should have made more clear in the demonstration. What about the off step? I'm going to draw the 3 graphs for the off step. Signal is on and goes away. Y was high. It was high. By the way, I didn't say that, but I'm assuming that the other signal, SY, is present all the time. So all of Y is Y star. When it's made, it's active. And of course, you can analyze what happens when you have steps of SX, SY, but I'm not going to. You can do it now, but I'm not going to do it for you. You can do it. So when I turn off X, Y starts decaying, because it's degraded and removed. What about Z? Yeah. When there was a signal, Z was on in steady state. Now we take it away. It only takes one arm of the AND gate to go off. So Z transcription stops immediately, and Z decays away by removal. And you see, there's no delay. So this little circuit does what we call sign-sensitive delay. Sign-sensitive, it's sensitive to whether it's an on-step or an off-step. Yeah? Yeah.
Explain again what's the difference between Y and Y star, but what is the process that turns Y into Y star?
So the question is, what's the difference between Y and Y star, X and X star? These protein transcription factors are little machines. They're built to be in two molecular states. They switch on a microsecond time scale. When something happens, when they're designed to bind a signal, like a sugar molecule, like a ribose, and that stabilizes one state. That state has a chemical structure that allows it to bind the DNA letters. So X star is the state of the protein that makes it possible to bind the DNA and activate production. Did I answer your question?
This is not part of the transcription translation. This is something additional. When you make a protein X, you need also to activate it.
Yeah. Yeah, almost all proteins, a lot of proteins are built to be in just a finite state of molecular states, like state machines. And most of them aren't active all the time. They need to be activated by phosphorylated or bind something or something. There's a button that needs to be pressed. That's the way proteins are.
And these SX and SY are different proteins or substances?
Well, in the case of the ribose system, cyclic AMP is a small molecule that signals for starvation, and this SY is a ribose, which is a sugar. So these are small molecules. And in other cases, it's a protein comes and phosphorylates, which means adds a phosphoryl group, which is charged, and that affects the conformation. Sometimes it's a protein partner that blocks you, and only when it's phosphorylated you become active. There's many ways to activate. So this is the mechanistic level of biology. This is a huge amount of work on each protein, how exactly it's activated or not. And in this course, we try to work on a more abstract level, because these things happen on the fast time scale. I don't care how it happened exactly. I just needed this logical.
To our purposes, you never write an equation for this Sx and
Right.
Sy. You assume that everything
Yeah. I don't write an equation for x, and sy, and because they're on the fast time scale, and assume therefore that things happen just like instantaneously. But if I wanted to go into the second time scale, I would need to write equations that look very much like the equations I'm writing, rate equations, and you get these exponential convergences just as they happen so quickly. They happen so quickly, they're within the thickness of this line, because there's orders of magnitude temporal separation. Are you satisfied? Okay. Thank you. And all these questions are important, because if you have a question you don't understand, there's guarantee there's other people who don't understand. Yeah. Of course, at the same time, there's other people who are groaning because they know this because we have these two populations here, two cultures, biologists and not biologists. We need patience, understanding, compassion for each other. On step, delay, off step, no delay, sometimes with delay, experiment. So Shmulik Mangan wanted to test this out. So he took this system and he glued green fluorescent protein here. And he added steps of starvation, like adding cyclic AMP. You can add that actually outside the cell. Or take away glucose to make the cell starve. So on steps and off steps. And he compared it to a perfect control system that he had, which is just simple regulation, no feed-forward loop. There's a lac repressor here for the experts. He neutralized it with IPTG. It's a different sugar. It's a different sugar gene, okay, with no feed-forward loop. And what he found was that with the on-step of cyclic AMP, okay, he glued GFP here too. These are two different cells. You compare them. This is the production of LAC-Z with no feed-forward loop. And this was for the Arab B, AD, feed-forward loop, which is a delay of about 20 minutes in this system. For the off-step, the GFP decayed away in both cases with no delay. So that's, for example, a way you can test what's going on with inside the living cells as they behave. Now, why 20 minutes? So it turns out that E. coli has these spurious pulses of the signal cyclic AMP when it transits between growth conditions that last about 20 minutes. So the purpose of the circuit is it's costly to make proteins. If you don't want to commit to making the system, you want to filter out. If you want to filter out pulses that are shorter than this delay, this circuit is good. But of course, when this signal is gone, you want to stop making protein right away because protein is costly. It's an asymmetric situation. You want to get benefit from the proteins, but they're costly. So on and off are different. So engineers also use sign-sensitive delays in situations like that. For example, in the elevator, there's a ray of light that opens the door. When you suppose you put your hand in, you take it out for a brief pulse of time, nothing happens. Take it away for a long time, doors begin to close. You put it back in, they immediately open. So it's an asymmetric situation. You don't want a person to get stuck in the door, and you're willing to pay that annoying delay to make sure. Let's take a nice deep sigh of relief. The reason I do this, by the way, is because when somebody comes in, you notice it, and it's good to bring things like that to the level of the group consciousness. Everyone feels better, I hope. And it's nice to breathe. So, Coherent. So we think that evolution, again, put this sign-sensitive delay in these hundreds of systems whenever you need to protect against small pulses of signal. You only want to, basically, you can call this also persistence, atmada detector. What about, so we talked about the AND gate. What happens when there's an OR gate? What happens when there's an OR gate? Okay? Instead of an AND gate. That means either X or Y can bind. Okay, I want you to think about that for a second. Think about that for a second. What happens when there's an OR gate? And now turn to somebody sitting next to you or behind you and discuss with them what happens when there's an OR gate in terms of this on and off steps. Say hi to the person sitting next to you and discuss with them. Yeah, you can talk with her, or okay, what's the answer? What's the answer? What happens after an on step? How many people think there's a delay after on step? Who thinks there's no delay after on step? Who thinks there's delay after off step? Who thinks there's no delay after off step? Okay, so the answer is there's again sign sensitive delay, but it's the opposite sign. It's the opposite sign. There's no delay after an on step, and there's delay after an off step. It's the opposite. Why? Because after an on step, Y star starts being made. But because it's an OR gate, Z immediately starts being produced. Because of the OR gate, X star is enough. No delay. But after an off step, Y, of course, goes away. Z doesn't have X star anymore, but it has Y. Thank goodness. So Y is still there to support Z. When does this stop? When Y goes below its threshold, its repression threshold. At this point, Z starts going down. So we bought ourselves an extra amount of time. How much time did we buy ourselves? Y steady state, e to the minus alpha t delay equals kyz. That's the equation for what this time is. This t delay. What time? Because it's an exponential decay. And the answer, and you don't have to write it down or be worried about it. Don't be worried about it. I said don't be worried about it. Is what? Y steady state divided by kyz. Something like that. So the point I want to make here is you can tune this delay by playing with the parameters of the protein, the removal and production, the removal rate of protein Y. It's steady state. The higher steady state, the longer this delay. The bigger the threshold, the smaller the delay. So you can tune it. Evolution indeed can tune these parameters by mutations that change Y steady state, Ky in order to produce the correct delay. But whatever you do, because there's a gate, AND or OR gate on a delayed and the direct path, you always have a sign-sensitive delay. You can't get around it. That's a property of the feed-forward loop. As long as there's two steps, which are slower than one step. Of course, you can have two steps that are faster than one step if you make huge degradation rates or it's microRNA or something like that. But for transcription factors, that's the situation. So you switch the OR and the AND, you switch the sign-sensitive delay sign. An example. So E. coli, when it's starving, it says, no more. I'm going to go away. I'm going to make an electric motor that spins at 100 hertz, that rotates a huge propeller called a flagella that's 10 times bigger than myself, and I'm going to swim away, and also a little computer called the chemotaxis system, which we'll learn about, to tell me where to go, up gradients of food. Isn't that amazing? And to make that, it does it in stages. And it takes it actually 2 cell generations to make the motor and the propeller. So it's a long process. It's a multi-step process. So it makes a motor. And when the motor is ready, it exports a protein called Fly-C to make the flagella that rotates. It makes a lot of these motors. And the circuit is this transcription factor that senses a tough situation activates this sigma factor, and together with an OR gate, they activate the protein that makes the flagella. This is also positively autoregulated. A motif within a motif. By the way, the signal to fly A is motor is ready. How does E. coli know that the motor is ready so that it can export the flagella? This is an amazing design. There's this inhibitor of fly A that binds it and stops it from making a transcription factor. And it's built so that when the motor is ready, it's secreted out of the cell. So only when the motor is ready, fly A becomes active. And then you start making the flagella, the flagellin, and that's also exported out of the motor and makes the monomer after monomer, makes this tube. The monomers go from inside the tube and go to the outside and build this flagellum. Okay? It's a wonder of biology. It's like taking some Legos, throw them in the air, and they land out in like a castle. I can't stop being amazed. So again, when you do this GFP situation, and you add the signal and take away the signal, you see that there's no delay when you add signal, and that there's a delay when you have the off step. If you delete fly A, or even delete its binding site in this promoter, you can get rid of this delay. The delay is about one cell generation. So what does this delay do? It means that you're swimming, you're out of the tough situation. You stop making the motors, but you still invested in a motor, so you also invest another generation in making a flagellum. Complete what you've started. In engineering, it's like an uninterrupted power source. The power goes down, but there's a battery hooked up to your computer that keeps it running for another hour or something. So that's what the coherent type 1 feed-forward loop with an OR gate can do. I'm so proud of us. We can take a nice deep sigh of relief. We have a lot of work to do in the second half, so please use the break to refresh yourselves, to come back ready for more. We come back at 10:10, and I'll see you soon. So what's the purpose of this positive autoregulation here? He asked. Nice deep sigh of relief. Positive autoregulation can increase the delay and plays a role in increasing this time. So here's an example of a motif within a motif with a function that you can understand on its own and has an additional context when it's inside a bigger piece. By the way, we think here about these 3-gene circuits in our brain, but inside the cell, of course, they're connected to many other interactions, non-transcriptional, post-transcriptional, and transcriptional. So you can ask, how dare we talk about a 3-gene circuit and talk about a function when, in fact, it's part of a much larger network? That's why it's important to do, let's take a nice deep sigh of relief, experiments where the feed-forward loop is inside the real living cell. And that's the experiments show, so far, in all the systems studied that I know of, that this function of sensitive delay works inside the cell. So somehow it's embedded without breaking the autonomous function of the circuit. All right, last hour we talked about the feed-forward loops, the coherent and the incoherent. The coherent type 1 feed-forward loop with AND gate, sensitive delay, a delay after an on step, with an OR gate, sensitive delay after an off step, giving you some extra time when the signal goes away. What about the incoherent feed-forward loop? That was kind of puzzling to us. And Shmulik Mangan suggested some functions for it. So let's look at the incoherent. Incoherent type 1 feed-forward loop is a pulse generator and response time accelerator. What do I mean by that? Suffrage. Oh, by the way, there's a CIS biology retreat next week, and next Thursday in particular. Who's going to be at that retreat? 1, 2, 3, 4, 5, 6. Okay, so we are going to have a course next Thursday. Next Thursday. Yes, there'll be a course. I'm going to come back from the retreat. And those of you that are in the retreat, you can also consider coming back. And if you don't, we have it, everything is online. Oh, yeah, sorry. Yeah. Who's going to be at the molecular cell biology retreat? 2, 3. Yeah, okay, so we're going to have a course. Yes. Yeah? All right. Incoherent type 1 feed for loop is a pulse generator and response time accelerator. So let's take a look at that. This is the signal. We do our little plots. This is time. This is X star. This is Y star. And this is Z. But in order to proceed, we have to understand what it means for an activator and a repressor to work together at a promoter. What does that do? So the typical situation is that you have, if X star is bound, but Y star is not, you have the Z production rate is some rate, high rate beta. So that means when X binds, but there's no repressor, that's high transcription. When the repressor binds, it spoils the party. When Y star binds, you go to a smaller production rate. All right? Sometimes you can go down to 0 if it's a really strong repressor. So that's the input function we'll work with. So let's take a look here. We have an on-step of signal. Y starts being transcribed, translated, produced, accumulate. So when X star binds, we have a nice production of Z. If there was no Y star, if we delete it from the chromosome, for example, it would like to go high to its steady state. But Y crosses its activation threshold. And at this point, Y transcription stops and goes down to a different steady state, which is smaller. Maybe down to 0, depending on beta prime.
Does it have to go down, or maybe it will go higher but lower than beta over alpha?
So Asi is asking, does there have to be an overshoot? There doesn't have to be an overshoot. If it so happens that if this crossing point happens here, before it reaches its new state, it'll change from a fast slope to a slow slope. So it doesn't have to be an overshoot, but still fast, slow. Did I answer your question? So that depends on details. So for the right parameters, this forms a pulse, an overshoot. Okay, great. And a lot of times it's really a strong pulse. But I want to also make the point that this is also speeds up the response time. Why does it also speed up the response time? It speeds up the response time because if I make a simple regulation with the same steady state, beta prime over alpha, and the same removal rate, right? This is a mathematically controlled comparison. Its dynamics would look like this. The blue line. And we want to understand the response time, we look at this z steady state. Did I write here x? I meant z steady state, right? I want to look at how, when we get to halfway to z steady state, and you see that the response time of the simple regulation is bigger than the response time of the incoherent feed-forward loop. Simple regulation, so incoherent feed-forward loop speeds the response times, no matter if there's an overshoot or not, because as I answered you, it always starts at a fast beta and then goes to a slow beta, let's say. So it's always above the slow beta curve. Any questions? You really need good eyes to see what I drew there, no? This is feed-forward loop. And this is simple regulation going to the same steady state with the same alpha. This is the steady state. So we calculate the response time by the half steady state. This response time is larger than this response time. Why? Again, it's the same idea. You can drive fast if you have good brakes. You use the big beta to start, and then you activate a repressor to stop. And what's the advantage? So let me just, we can do here a huge meta review, a super review for all the 3 lectures that we've done so far now by noticing that we now have learned about 3 ways to speed your responses. Way number 1 is to increase alpha in simple regulation. Increase removal. It has this cost. You also need to increase production. The second thing is negative autoregulation. Right? With the same alpha, with the same removal, you can get faster response times. But the problem is that this works only for transcription factors. That is to say, you need to be a protein that knows how to reduce its own rate of transcription. But if you're some machine, some flagellin or something, you can't do that. And then we have the incoherent feed-forward loop. It works for all target genes. Z. Okay. I just want to also say that all 4 coherent feed-forward loops are capable of sign-sensitive delay, and all 4 incoherent types of feed-forward loops are capable of response acceleration and pulse generation. If you're interested in understanding that, you have the tools to figure that out already by making these little sketches. You can do it. Who knows? Maybe in the exercise you will do it. Yeah, okay, great. So, incoherent time pulse, speed up, the 3 ways. We're making very excellent progress. We're going to finish all the network motifs in E. coli. You're going to know how to calculate the dynamics of anything you want in E. coli. Why didn't you say don't erase? Why didn't you say don't erase?
Don't have a fast response time.
Yes, good answer. You don't have a fast response time. The 3 ways, which by the way are in the book, I'm not making anything up here. Of course, I wrote the book, is to increase removal rate in simple regulation, to have negative autoregulation, which also works together with increasing removal rate. They can work together beautifully. So negative autoregulation, and incoherent type 1 feed-forward loop. 3 ways to speed your responses. And they work together. If you think about it, if Z has negative autoregulation, it's also at the bottom of an incoherent feed-forward loop with a really fast removal rate. You can get a mean fast response time. Okay. I really want not to erase before you guys are finished. That's my ideal. So help me, okay? Say, ah! Okay. So I want to say that in E. coli, there are only 3 major families of network motifs. I'm going to say 4 because it depends how you define it. There's this, right? There's this. So I'm not going to take care of the signs right now. So autoregulation, feed forward. There's this. We're going to talk about right now. So autoregulation, FFL, single input module, and this one. Interesting. So we've covered this so far, and we're going to talk about these 2. This is a generalization of the x to z1, x to z2, this kind of pattern that we saw exist in E. coli. This is just a transcription factor. It regulates a bunch of genes. The non-random thing about it is that there's no other arrows coming in. It's an isolated module. A lot of times there's also autoregulation. So x regulates z1 to zn with no other inputs. So there's no y that regulates any of these genes. It's a module. What does it do? Single input module. For example, the arginine repressor negatively autoregulates itself. When the amino acid arginine is around, it's active in repressing the genes that make carbon atom by carbon atom the amino acid arginine. So they produce actually arginine, closing a fast feedback loop. This circuit makes sure that if there's no arginine, you don't make the proteins that make that amino acid. And if there's no arginine, you turn them on. And always these genes have a common biological function. They're a pathway, like a pathway that makes some chemical. They could be the proteins that make up the flagellum motor. They could be whatever. But there's also a delicate, but the single input module also has a dynamical function. It can generate temporal order. First gene, second gene, third gene. And how does it do that? So the master transcription factor binds these different genes. It has a binding site in the promoter of each of these genes. But these binding sites can be a little bit different. They can have differences in them, which determine the chemical affinity of the transcription factor of that DNA. And it could be different. So you can have different thresholds for different genes. X can have different thresholds. Ki for each gene. And therefore, check it out. Okay. We're going to need a little bit more. Maybe I'll do it here. I'll do it. So this is X star. Now, instead of what I did last with the feed-forward loops, where we had a abrupt, like on steps, I'm going to let X star here change gradually. If X star changes gradually, it crosses the different thresholds at different times, right? And therefore, This is the first downstream gene. This is the second one. That's as simple as that. Indeed, in experiments, on pathways, like this arginine system, you can see what we call just in time production, where genes or proteins, you can say, proteins are made in the same order as the pathway. So to make arginine, you take some molecule, the enzyme ARG-A changes some carbon atom, the enzyme ARG-B adds an OH group, the enzyme ARG-C cuts something off, etc., until you get what you want. That's the magic of metabolism. The thing is that there's a temporal order encoded into the DNA, setting the Ks in such a way that the order that when the cell needs to make arginine, it turns on this one first, and this one second, this one third, etc. That's called just-in-time production. Why? It takes some time for A to make S2, which is a substrate for B. So if you make B too early, you're wasting proteins. You're making unneeded proteins. So this protein cost, we think, supplies a pressure not to make a protein before you need it. Questions?
When you compare this mechanism for
Temporal order
to having S2 induce the production of RNA.
So the question is, you can design a circuit where only when this is around, there's a transcription factor that activates the next one. Yeah. So that's another alternative design, and we can think about what are the advantages and disadvantages, but certainly what it means is you need a transcription factor for each step. And that's a big cost. Okay. And also, the time scales here are really, the delays here could be of minutes, and transcription factors are a little bit slower than that. It is the case, by the way, that arginine itself inhibits usually by binding the first enzyme. So there's this kind of fast-acting, what's called feedback inhibition. It's not transcriptional. It has to do with protein level activity, and so the product inhibits the first step, and there's also engineering reasons for that. I'm not going to get into it. Okay. Yeah.
When you say there is a threshold, but they do start to produce before a little bit, right? To make it not linear.
Okay. So what Ashley is asking is about the input function. So the ZDT is some function of x star minus alpha z. This function here, I'm assuming, is a step function, only when x star is greater than k. But if it's not a step function, it's a Hill function, let's say, you get a little bit like this.
In biology, it's more like a new function.
Yeah, in biology, there's no real step functions usually. It's usually the Hill function I talked about before, which is, but it could be quite steep or not. But it doesn't matter what the function is. You still get a temporal order of, let's say, the halfway point. No matter, as long as it's an increasing function of x. Yeah. So that's, again, I'm playing here in this course on several levels of approximation in order to be able to explain the intuition of it. Yeah.
Throughout the course, you also assume that, I guess, metabolic changes happen at a very rapid time scale. So wouldn't the argument of the just-in-time transcription assume that this happens at a slower scale?
Okay. Right.
Why is
Yeah.
this in this case?
The question is, what's the time scale of a delay between two metabolic steps in a metabolic pathway? So far, we only talked about transcription. Here, I'm talking about this chemical factory where E. coli makes itself by carbon atom called metabolism. What's the time scale between two steps there? And I also thought it was very fast until we did some calculations and looked at some papers. And it turns out that if you have, let's say, if you look at how much substrate you need for this to work, so So the typical KDs could be millimolar, which means 1 million molecules per E. coli. And then you consider this enzyme is present, let's say, at 1,000 copies and works, let's say, at 10 times per second. You need, if I'm not mistaken, 100 seconds, which is a minute. So you can get to minutes delays in the activation. And that's why it's so useful not to have, a cell generation time, but something that can give you minute delays. Does that make sense? Now, it's incredible that saving a protein for 2 minutes, not making it only a 2-minute delay, has a selectable advantage. And we think it does because of the selection is huge. If you're more economic in E. coli, you win this race against the other bacteria that are growing exponentially. Make sense? Yes?
I think maybe it's not just a matter of economy. Maybe the protein is actually damage the cell somehow if the cell doesn't use it, just to get it around.
Yeah. So in some situations, having a protein without having the previous step, let's say, for example, could be damaging. So you can think of let's say, like building the motor, flagellum motor. By the way, flagellum motor also has, I think, 14 proteins timed, making first a ring inside the cell, then something goes through the first membrane, then a shaft, then a ring going through the next membrane, then a flexible hook, an adapter, then that anti-fly flies out, and then the propeller. So all these steps, you can't, if you just make those proteins without the previous step, they'll accumulate inside the cell and be damaging. So that's another example of toxicity. You want to avoid toxicity by only making the protein just in time. Good point. Yeah. Listen, that's very good questions. Can I erase now? Anyone have any objection? Okay, I'm going to erase. So we're really going to figure out all the network motifs in E. coli. Single input module. When you look at higher order patterns, they talk about 3-node patterns. This is, of course, can have many genes. But essentially, it's based on this 3-node pattern. If I look at 4-node patterns, 5-node patterns, if you look at 4-node patterns, there's 199 possible subgraphs. I'm not going to draw them all on the blackboard. But you ask which one is a network motif, occurs again and again and again, and far more often. Out of 199, there's only 2. So out of 199 4-node subgraphs, only 2 are network motifs. It's this one, and this one. This is multi-output feed-forward loop. And when you see this, you understand exactly what it does. Because if this is a coherent type 1 feed-forward loop, it does it for Z. It has a sign-sensitive delay for Z1. If this is an incoherent type 1 feed-forward loop, it has a pulse generator for Z2. You understand it. But it's interesting that feed-forward loops could have been glued in different ways together. You could have had 2 X's controlling the same Y and the same Z. This is another way to glue together feed-forward loops. It's also a 4-node subgraph, but it doesn't happen. So this is the way E. coli glues together feed-forward loops. When you look at neural networks, I want to say, you find essentially the same motifs, but neurons are different from E. coli in gluing together feed-forward loops in this way a lot of times, like 2 sensory neurons controlling an interneuron. And this bifan, this is like 2 inputs, 2 outputs. You have to understand the logic gates. This generalizes to a large family. So I'm going to write x1, x2, x3, x4. You get these big guys here, dense, overlapping regulons. And we don't really understand what to say about them, except that you need to understand the input function.
Isn't it a good way to correlate the expression of a few genes that are like related, but not necessarily will have to occur at exactly the same time?
Correct. So what this dense overlapping regulon does, it's like what's called how's it called in electronics? We have a lot of logic gates. Who's from electronics here? Gate. It's like a gate array. You have inputs, you have outputs, and you want to combinatorially regulate the outputs. So you can have, let's say, some stress proteins, and these ones respond to this stress. This one responds to stress A and B. This one responds only to stress B. This one responds. So you need to know the logic input functions. Actually, that's part of the biggest, least known part in biology is how. So we know a lot about the arrows nowadays because there's experiments in biology where you can discover an entire gene regulation network in one. It's not even a PhD thesis. It's like half a PhD thesis. Whereas, let's say, when I was a postdoc 15 years ago, it was a PhD to understand one arrow. Now, it's within a PhD to understand an entire, to first approximation, gene regulation network of a cell. That's 10,000. It's an increase of experimental power by a factor of 10,000 in 15 years. And there's no indication that this is stopping. Biology is now exponentially increasing in experimental power. So the amount of resolution of knowing things about the cells is growing at a breathtaking rate. And that's why systems biology now is such a hot field, because we'd like to understand this information. And why did I say that? Because what we don't know still, and we need technologies for that, is to understand those input functions. Is there an AND gate when there's three arrows? Is it X and Y or Z? Is it X exclusive or Y and X exclusive or Z, or whatever? We don't have the technology, I think, and so that's a technical challenge if anybody's interested. And I want to just say that we can now talk about, now that we know these, and that's it. Those are the only subgraphs. There's no other subgraphs. So as you see, all these are directed subgraphs. You call it like a directed acyclic graph, for those of you from computer science, but with much fewer subgraphs. The diamond, for example, doesn't appear in E. coli transcription network. This doesn't appear. But it does appear in other kinds of networks. So I didn't tell you what this does because it's just a series of logical steps. You need to understand the details of it. And can I erase? Anybody have objection? Objection overruled. So huge simplicity out of a number of subgraphs it could be. And so integrated circuits made of motifs. How do these motifs fit together to make the entire network? It turns out, again, you can make a picture of the network, building a big graph with all the arrows. That's called usually in our field by the technical term fuzzball. It looks like a huge mass of arrows, and you can't understand it. But if you look at network motifs, you can make a coarse-grained description by drawing, let's say, a rectangle where there's a dense overlapping regulon, a triangle where there's a feed-forward loop, a square where there's a single input module. And then you get a picture. It's quite complicated, but you can understand it. So the E. coli network has a layer of dense overlapping regulons. So transcription factors regulating. Inside here are the feed-forward loops, where you have X regulated G, Y regulated as gene, and X regulates Y. So also you have the single input modules, where you have one regulator regulating a lot of these. That's the way it looks. There's no dense overlapping regulon at the output of another dense overlapping regulon. A single cortex where the distance between the inputs, the transcription factors, and the output genes is usually 1 step or 2 steps in the feed-forward loop. In the world record, there's 1 circuit we know of in the flagella system that has five steps. That's rare. So there's no cascades. X activates Y, Y is Z, Z double. Why is that? Why is it so shallow? You can say, why is it? We think again, it's an engineering reason. The time scale for information to go down each transcriptional step, as you know by heart already, is on the order of a cell generation time. And E. coli needs to respond fast to nutrients, stresses. It needs to respond now for itself, not for the children, not for the grandchildren, for itself. So the part of the computation it does with transcription is limited by the physical timescales of the system. There's other kind of networks inside the cell that we'll discuss in the next lectures. Phosphorylation, where you don't need to make new proteins. You use already the existing proteins, you modify them chemically. They work much faster. And there you do see cascades, you do see additional network motifs. But not in the transcription network of E. coli. All right? Yeah.
How accurate is it to use E. coli as a toy model for other cellular systems? Like we say that cascades don't exist in E. coli, whereas we know that cascades are very dominant in human cells. So, like, where does it stop?
Okay. So the question is, these network motifs, what happens in other organisms, right?
Yeah.
So E. coli and another single-cell organism, yeast, have the same network motifs, but they're completely different kinds of proteins. So it's not that E. coli network motif was conserved over evolutionary time and became a yeast network motif. It's different transcription factors wire up in the same ways and don't wire up in the same ways. See, there's no cycles there. So that's the universality for these single-cell organisms. For let's say animal cells, human cells, there's 2 kinds of gene regulation networks. Gene regulation networks that respond quickly to signals have the same motifs. So let's say recently, 2012, a huge project to map the gene regulation network of many different cell types in human found that the feed-forward loop, coherent type 1 and incoherent type 1, are the hugest network motif in human cells. And then there's also what's called development, which is the problem of going from an egg to a baby. Diaper. I have a baby now, 1-year-old baby. And there you need to make slow, irreversible decisions. You go from a stem cell, let's say, to become a blood cell. Oh, maybe it'll become a red blood cell. Or maybe I'll become a muscle. Maybe I'll become a neuron. So this slow. And there you find these network motifs plus additional. So there's additional network motifs in developmental gene regulation networks. And they are the cascade, a lot of times a cascade of repressors, and positive feedback loops. They're either double positive or double negative. So these are 2-node motifs. And these things function like memory devices. When you activate X, there's no Y. When you activate Y, there's no X. So it's an either or. Or when you activate X and Y lock together, and forever you're a blood cell. Forever. Until Yamanaka comes and reprograms you. That's a different story. So these motifs exist in all animals, in all cells. And if there's a developmental process that's irreversible and slow, then you can use the slow time scale to actually make a clock of cell generations, if you want.
So is it safe to say that there's like unicellular network motifs and multicellular?
You could say that, yeah. And of course, in the multicellular cells, our cells have some part of them are like unicellular.
Right.
And then the part that's multicellular to make all the patterns and the division of labor has its own motifs. So I know I answered your question, right? I'm so satisfied. So thanks for this question. Wow, we're so good here in this class. I'm very, very proud of you guys. So usually I do this in 2 lectures. Here I'm doing it in 1 lecture. And how are you feeling? How many think it's too fast what I'm doing? It's fast? Yeah, so raise your hand if it's too fast. So we need to know for the next lecture, right? Don't be afraid. How many people think it's too slow? How many people think it's the right speed?
What was population?
You want to see sporulation?
No, you had it on the list of population.
Where? Oh yeah, I did say I didn't talk about sporulation. That's right. So I'm going to do it now because we have a couple of minutes to do it. Okay, so what should I erase? I'll erase this part, okay? I told you that you'll be able to calculate the entire, like any gene regulation network dynamics. And I want to prove it to you by looking at the different bacteria. It's called B. subtilis. It lives in the ground. It's very nice. And when it's starved, it makes a spore, which has its DNA, and it's indestructible. It can live for decades in the ground. And you put in good conditions, it grows back again. It's like a seed. And to make the spore, the mother cell doubles their DNA and starts making these waves of gene expression. Each one coats the DNA more and more and more. And then the mother commits suicide, and the spore is left. It's a touching story. And the transcription network that regulates this process was figured out by the Losik lab at Harvard, and it looks like this. So it has an incoherent type 1 feed-forward loop hooked up to a coherent type 1 feed-forward loop hooked up to a second incoherent feed-forward loop hooked up to a final coherent feed-forward loop. So this is an I1, this is a C1, this is an I1, and this is a C1. This is a starvation signal, and this is a signal that comes from other cells around. Here, it's actually tens of genes. So this is a multi-output feed-forward loop with different Ks allowing for temporal order. Okay. So based on this, on our course, you know the dynamics of this complicated-looking circuit. This is time. These are the genes. So when there's starvation signal, the incoherent type 1 feed-forward loop generates a pulse. It also makes X2 after a delay. Okay? Because of if there's other signal, this one generates another pulse. And a final delay, based on this coherent feed-forward loop, generates the last wave. So it's a 3-wave pattern. These Z, I told you, are many genes. So with the different Ks, you can have different temporal orders inside here, inside each of these waves, like the single input module. Same idea. And the feed-forward loops here are integrated to give you this temporal pattern. In fact, you can prove universality. You can get close enough to any temporal circuit you have, provided you have enough coherent and incoherent feed-forward loops. So they're a universal circuit. They're better than feedback loops because they don't go unstable. They're feed-forward, so delays and noise never make them unstable like feedback loops. They're a great way to work with noisy components to get time courses. So we look at this class, what we did today, we looked at the feed-forward loops as sensitive delays, pulse generators. We looked at speed up, the 3 ways to speed up, single input module with its temporal order and just-in-time production, dense overlapping regulons, the global structure of the E. coli network. We talked a little bit about network motifs in development, which are additional network motifs in animal cells. But again, a tiny amount of circuits, as far as we can tell, describes this, what looks quite complex graphs, the gene regulation networks. And each one of these circuits has candidate functions. So when you discover it in your biology work, you can have a hypothesis for what this circuit might be doing. And of course, you need to do an experiment to see if it actually works that way. Maybe it's wired into the cell in a way that somehow interferes with this function, or maybe it's part of a larger circuit. But at least you have a hypothesis or a language to talk about the brain, you can say, or this part of decision making inside the cells. So thank you very much. Let's take a nice deep sigh of relief, and I'll see you next week.